Cloning contains a few different processes that produce identical genetic copies of a specific biological entity. It's the copied material of the same genetic makeup as the original that is referred to as a clone. Cloning can happen both naturally and artificially in a lab. Natural cloning happens in nature within single-celled organisms, like some plants and bacteria, through asexual reproduction. Asexual reproduction, the way in which bacteria is able to reproduce, generates a new individual from copying one single cell from the parent organism to carry on their genetics. In humans and other mammals, the most common scenario to find cloning happen naturally is in identical twins, though triplets and quadruplets are examples as well. While twins have a genetically different makeup as either parent, they have nearly the same genetic makeup as each other due to the fact that they are produced from the splitting of one fertilized egg. Artificial cloning, there are three main types: gene, reproductive, and therapeutic cloning. Gene cloning, also known as DNA cloning, produces copies of genes or small segments of DNA. The procedure consists of inserting a gene from one organism into a genetic material of a carrier such as bacteria, yeast cells, or viruses. DNA sequencing is an important step in being able to understand the genetic makeup of an organism by helping locate regulatory and gene sequences, compare homologous genes across species, and identify mutations. Knowing the sequence of a particular gene will assist in understanding the function of the gene. There are two ways to accomplish reproductive cloning, the most controversial way being somatic cell nuclear transfer cloning. This is the process of cloning. The entire genetic code, except for the mitochondrial DNA, is produced from a single body cell of an adult individual. The nucleus is removed from the egg, leaving the cytoplasm of mitochondria. Then a body cell is taken from an adult individual who is to be cloned. The DNA is extracted from the nucleus and inserted into the prepared egg. Then the new cell is then induced to divide using either chemical or electrical stimulation, thereby commencing the development of an embryo. After several days, the dividing embryo is then placed into the womb of the recipient and allowed to develop to term. The result is a clone. The most known story of this is Dolly, a sheep born in Scotland in 1996. Dolly was one out of 277 cloned embryos to be born alive. This very low efficiency, combined with safety concerns, presents a serious obstacle to the application of reproductive cloning. The other way to accomplish reproductive cloning is through embryo splitting. This most commonly occurs within in vitro fertilization, or IVF, when women have trouble conceiving a pregnancy. This process brings together a man's sperm and a woman's egg outside of the woman's body to generate a zygote.
Oh my.
The last form of cloning is therapeutic cloning, which involves creating a cloned embryo for the sole purpose of producing embryonic stem cells with the same DNA as the donor cell. These stem cells can be used in experiments aimed at understanding disease and developing new treatments for disease. Since stem cells can continuously regenerate like cancerous cells, there have been many in the scientific field who oppose therapeutic cloning.